And our next speaker is here with us today. So Dr. Kacie Deters is a neuroscientist and an assistant professor at the University of California, Los Angeles Brain Research Institute. Her research focuses on predictors of racial and ethnic disparities in cognitive decline and dementia. Today, she's going to speak about identifying risk factors in the Black community. Dr. Deters.
All right, hi everyone. Thank you so much for the introduction. So I'm Kacie Deters, assistant professor at UCLA. I am brand spanking new. I've only been a professor for about 8 months. So I'm actually going to be talking about some research that I did during my postdoc, and I'll be talking about some of the directions that my lab is will be going, but not sharing any data yet. I'm still working on it actively. So. Also, I want to mention, I've never been here before. Very beautiful. I got here a little too late and I was pretty hungry. So I'm really looking forward to exploring these gardens that Dr. Summit was talking about, maybe tomorrow during my lunch break. So if you have any other recommendations for me to explore this campus, please let me know. Yeah, sure. I'm also going to be talking about not just only risk factors, but also biomarker trajectories. So some of you here may be familiar with this biomarker trajectory chart from Dr. Risa Sperling, who's over at MGH. And so this was really, great model of all of these different tools that we have to analyze different biomarkers using a bunch of tools, CSF, PET, MRI, clinical function, and cognition measured through neuropsychological assessments. And I've been focusing, actually all of my research today has been using secondary data sets. So I've been using data sets like ADNI, A4, Rush data sets. And the really great thing about using these secondary data sets is that a lot of them have been followed now over time. And it's been really great because a lot of these biomarkers have been captured in these different data sets. And so I've been looking at a lot of clinically normal, the preclinical stage, Black older adults, and focusing on trying to identify early biomarker trajectories. One of the things that I learned though through my training is that, a lot of these trajectories, risk factors, a lot of the tools, a lot of the cutoff points, everything has been created and validated in the white population. And so I was really curious once I started to investigate this, I started to notice some of the differences in these risk factors and trajectories. And so that's really led my research up to date. And that is, really focusing on what are the risk factors, what are the biomarkers for Black individuals. Another thing that is different is, looking at genetic risk. So GWAS studies or genome-wide association studies have been used to identify a lot of the genetic risk for Alzheimer's disease. So this is the most recent GWAS study, again, highly focused on a European ancestral group. And there are a lot of genetic risk factors that we've seen to date, including ApoE, PCOM-CLU, BIN1. But again, my question was, considering that most of the GWAS studies have been focused on white individuals, what is the genetic risk profile for Black individuals? And this is really, really important because looking at a recent, or not so recent, but a really widely talked about study from a colleague of mine now at UCLA, Dr. Elizabeth Rosmeda, she has this really great research study that looked at dementia incidence rates by race and ethnicity from individuals that were seen at Kaiser Permanente in Northern California. And as you can see here, incidence dementia rates are on the y-axis, and different racialized groups are on the x-axis. You can see that people who identify in this study or in this cohort, Kaiser Permanente, as African-American or Black, actually have a higher incidence rate of dementia relative to all other groups, including the white group. So again, I want to center back on why I think research within the Black community is really important and not comparing. Again, talking about her research study that Black Americans are disproportionately more likely to have not only dementia, but also cognitive impairment relative to white individuals. However, whiteness continues to be centralized in research, including the genetic profile. This is a figure that denotes the ancestry catalog that's available for genetic studies, GWAS studies, and you can see by looking at the blue that represents the European population. Most of the genetic profiles that we have are from European, while if you look at the orange, I don't think that clicker works, but the orange is the African ancestry category, and you can see that only represents 2%. So how can we accurately, identify genetic risk when we don't even have an adequate profile for such a large percentage of our population. And again, those are disproportionately more likely to have dementia. This is extremely important because the US is becoming more diverse. For the first time, this is actually results from the US Census. And so for the first time, people who self-identify as just white in this country is actually on the decline, while people who identify as every other group is actually on the incline, the increase. And this was really interesting to me just in general, just because I'm Black and white. And so when I started getting into this research, everything is white, or it's Black, or it's Hispanic Latinx. And I was really interested about how do a lot of these risk factors actually relate to people who are multi-biracial. So talking about the first thing, so again, using markers to find in the white population and in the non-white population and why this may be an issue. So I really like using this figure from Dr. Weave et al. In a study using the Chicago Healthy Aging Project study. And so on the x-axis, you have global cognitive score. I'm sorry, on the y-axis. And so higher is better. And on the x-axis, you have age or time, if you want to consider it that. And then there are two groups racialized as white and Black. And we can see a few things from just looking at this one chart when it comes to looking at neuropsychological assessments. First, if we look at just baseline, which is that highlighted in green, so just one time point, you can see that white individuals have a higher global cognitive score or better relative to Black individuals. However, if you look at the slope of cognitive decline, you see that it's actually not different. And so this actually highlights two really important things when it comes to neuropsychological assessments that we use for, to define clinically whether somebody has Alzheimer's disease or dementia or not. And that is one, the racial bias that exists in our neuropsychological exams. I think we're all aware of this, and there are norming procedures, which is a very contentious topic, as we probably all know at this point. But it's something that I think, neuropsychological assessments are really the baseline tool that we use to assess memory, and we know about this cultural bias. And so it's one of those things that I do think that it actually does work, as we see by looking at the slope, to identify whatever cognitive domain the test may be referring to. However, if we look at someone's memory at one time point and it's a non-white group, there's this bias that needs to be addressed. And so also points out the need for longitudinal studies in Black cohorts. Genetic risk factors are also not the same for Black individuals. So I'm going to be talking about two genes on chromosome 19, ApoE, which I'm sure we're all familiar with, E4, greater genetic risk factor for Alzheimer's disease. However, something I did not learn until the last year of my PhD actually was that there are different frequency rates for ApoE4. So I have shown here, I think this is also using data from the Chicago Healthy Aging Project. You have people who, the different racialized groups on the x-axis and percent E4 frequency on the y-axis. Cognitively normal individuals are on the solid colors, and then those with dementia are in the patterned bars. And as you can see, Black individuals actually have a higher frequency of ApoE4. And this has been shown in a bunch of different cohorts as well, not just this one. So this one just has a really large number of subjects, so I like to show this data. But so yeah, Black individuals actually have a higher frequency of E4. So you would think then, if they have a higher frequency of E4, that this would translate to higher risk for Alzheimer's disease and dementia. But as you can see by the patterned bars, the patterned bars for Black and white individuals, there is no actual difference in the risk of the contribution of E4 to dementia risk. So although E4 is a contributing factor to dementia risk for Black individuals, there must be other factors, other genetic risk factors that are actually contributing to dementia risk in this population that we need to be accounted, or that we are not accounted for. And just to look at E4 a little bit more, there's a group out of University of Miami, Dr. Jeffrey Vance. I really appreciate and value their lab a lot. I think translational work is really important. And so although they do a lot of human association studies, they often take do iPSC cell studies. And so, for example, to look at E4 expression, they took people who self-identified as Black, had high African ancestry at the E4 loci, and plated these cells. Same thing with European local ancestry. So people who identify as white have really high European local ancestry at the E4. So And they showed that your E4 expression was actually lower. So purple denotes expression. And so you can see that if you have African local ancestry at E4 and your E4, you actually have lower expression relative to European E4 local ancestry. Another gene is TOM40, and so this is just upstream of ApoE. There's a specific SNP, I'm just going to call it 523 for short, that has been found to be associated with increased cognitive impairment and AD risk. So this SNP is associated with the poly-T repeat. So at the loci, it's like TTPPT, and you can see on the figure, there's a histogram of the number of T's that are represented in a particular group. So for example, the TOM40 length ranges from about 15 to 40 in this group. A lot of research has not been focused on TOM40 because there isn't variation in white individuals when it comes to this poly-T. It's if you're white, E4 and poly-T and have this specific SNP, you only have a small range of poly-Ts. So previously, as we know, science again has been very focused on European white individuals. It's just been, dismissed. Additionally, it is in linkage to equilibrium with ApoE, and so often inherited together. And so a lot of times people in the past just thought the effect of TOM40 was really just being captured from ApoE. And again, if we actually look at TOM40 in white and Black individuals, it looks almost identical. So again, you might think, okay, there's no variation, move on. However, some scientists earlier noticed that there were some ApoE and TOM40 haplotypes, such that if you are ApoE3 carrier, you have poly-T repeats that were less than 20 or greater than 30. And if you were an E4 carrier, you had poly-T repeats between 20 and 30. So we're going to call these short, long, and very long from now on. And if you look at the ApoE TOM40 haplotypes in Black, you'll notice Black E3 carriers very similar to white E3 carriers. But if you're a Black and E4 carrier, there's a much greater variation when it comes to TOM40 that was just never identified or assessed in the past. And so, and before we move on to those study results, you may be asking, where does this variation come from for Black E4 carriers? And I accidentally left this slide out, but this is such a beautiful example of genetic admixture that actually, if you focus on a European group and an African group, you'll notice that the European E4 carriers, as you see from this white E4 carrier group, that they contributed the long. But the African group actually contributed the short and very long. People who identify as Black in the United States, due to transatlantic slave trade, atrocities on our people who identify as Black here have highly admixed genetic DNA ancestry from African and European. So this Black E4 carriers that are focused in America is a great example of genetic admixture for this. So for this data, I used data sets from Rush University. I used 3, the religious order study, which looks at nuns, priests, and brothers from across the United States. The Rush Memory and Aging Project, MAP, Greater Illinois area. And then most of the Black individuals from this study came from the Minority Aging Research Study, or MARS, which is all Black individuals from Chicago and surrounding suburbs. And the PI is Dr. Lisa Barnes, who was also mentoring this project at that time. One of the great things about these Rush cohorts is they've been followed for like decades at this point, all using the same battery. And so there's a really rich neuropsychological battery that is done that I can follow cognition over time in these individuals. And so for this study that I'm going to show, I'm actually looking at global cognition, so the composite of all of the neuropsychological tests. So again, just to reiterate us to the TOM40, looking at the effect of this TOM40 variation in Black E4 carriers. For statistical sake, we had to group them, right? So we group these into presence of the short allele. So the colors should match the figure. So red is 2 short alleles, blue is 1 short allele, and gold or yellow is having no short alleles. So, and on the y-axis, we have global memory. So we can see straight off the bat that Black E4 carriers that have a short allele, so the blue and red, actually have a slower rate of cognitive decline relative to Black E4 carriers who do not carry this short allele. Now, if we look at this relative to the white E4 carriers, remember in the past, this hasn't really been investigated because when they did look at there's no variation. So it's really just capturing, it really is just capturing the effect of E4. So we can see two things on this figure. First, what I alluded to in the beginning, that if the cultural bias that exists in neuropsychological time test. So if you look at just one time point, that first time point there, the intercept, white E4 carriers have better memory relative to the entire Black E4 carrier group. However, if we look at the white E4 carriers compared to the Black E4 carriers who do not carry a short allele, the slope is also not different. There's no significant difference. However, if we look at the white E4 carriers relative to the Black E4 carriers who do carry a short allele, this Black E4 carrier group is also significantly better, showing significantly slower rate of cognitive decline relative to the white E4 carriers. So this suggests that these Black E4 carriers may be a potentially protected group. This was really interesting to me because when I did get into this research, I noticed that there was a lot of variation such that some studies on looking at the effect of E4 and dementia risk or cognitive decline in Black individuals vary. And so again, if we're just focusing on one genetic risk factor that actually may not be as important for the Black individual group, we may be missing specific measurements, risk factors. And just to also focus, again, looking at non-APOE genetic risk factors, this is a GWAS similar to the one I showed earlier, but this is actually done on just Black individuals. If you don't know, you do need a lot of power, a lot of subjects to run GWAS studies. And as we know, there is not a lot of Black individuals that have been recruited into our research studies. So it's been really hard to get like an adequate GWAS for genetic risk for Black individuals. And so there was one done recently. And actually this is the follow-up. I really appreciate this group because every few years, as we do recruit more and more Black individuals, they do update this GWAS, which I think is really important. And so they've identified, non-APOE genetic risk factors, both common loci, rare loci, and then some that have been previously reported. And I do want to point out at the bottom, ABCA7, that genetic risk factor, it's rare, but the loci they found. However, they did identify that this ABCA7 genetic risk factor actually had an equal or as great effect as APOE4 in this population. So again, we need to be focusing on other non-APOE genetic risk factors. Additionally, there was a GWAS that was done based on ancestral background. So this is looking at a GWAS focused on just African and people with high African ancestral background. And again, they actually identified two genetic risk factors, one that actually lowered AD risk for Black individuals, and one that actually conferred higher AD risk for Black individuals. So again, it's so important that we start focusing on research within the Black population. There's already great variation within the Black population. If we continue to compare our results to white individuals, there's always going to be disparity based on the cultural bias that exists in our tools. And I'm going to talk briefly, or have I already talked briefly? Yeah, talked a little bit about genetic ancestry. And so I really like to differentiate race and genetic ancestry because race is a social factor. There is no biological relevance. And I think, not only me, but a lot of people that are doing health equity research are trying to really make sure that there is no specific correlation between the two. It is very important that, we understand that when I use genetic ancestry, and hopefully when other people use genetic ancestry, that we are not trying to tie on a specific genetic risk factor with saying this is specific to, or this is like Black. Race can be important though, because as Dr. Klein talked about earlier about the importance of zip code and how it relates to redlining and specific cultural communities, it can capture important economic or epidemiological information. So, still collecting, a person's self-identified race can be important, but making sure that we use it as appropriately as possible is critical to equitable research. And then genetic ancestry, this is something that I am very interested in, and I have a KO1 that should be coming soon. that actually Dr. Prime is also a co-mentor on, so I'm very excited about that to look at ADI. I'm really interested in looking at the genetic ancestry factors at the specific loci and how those might help improve estimated or personalized risk measurements. Some notes about genetic ancestry, though, it is really correlated more with geographic location, right? You collect data, you collect blood and samples from people that are specific to a region, which goes to a question that I'm not going to go into at all, but this is really environmental, talking about DNA methylation up changes, but also there's a lack of genetic ancestry. Going back to that pie chart that I showed about the GWAS catalog, where only 2% were from the African population. We do need more representation of non-European populations. Another thing, just to go through very briefly, that's different. Post-mortem or pathological insults are also not the same. So Black individuals, I'm sure this is probably not news, but Black individuals have more comorbidities relative to white individuals. And so again, the field is so focused on pure AD pathology, tau and amyloid. When that is not the main feature for pathological cardiomyopathies in Black individuals who are diagnosed with Alzheimer's disease. Another thing is that, again, since we are so focused on amyloid and tau, it's interesting, there actually has not been a lot of work done in Black individuals to actually look at these differences, similar to the way that we just did with genetic risk. And so there's been a lot of variation in findings, especially in the amyloid. And so I actually wanted to look at amyloid in clinically normal older adults. So for this study, I used data from A4, which is the anti-amyloid and asymptomatic AD study. And so this is a secondary prevention trial, anti-amyloid study, and everyone had amyloid PET. So this was a great opportunity to actually look at amyloid PET, in vivo, and determine if there were any differences. So on the y-axis, I have continuous amyloid SUVR, and on the x-axis, we have APOE genotype and race. And so just right off the bat, just by looking, you can see that Black individuals have lower or better levels of amyloid relative to white individuals. But what was really interesting is that, again, E4 is supposed to be this genetic risk factor, but the lower amyloid was actually only significant in the Black E4 carriers. Again, suggesting here we have a well-established risk factor of E4 and amyloid, and both are showing different profiles in Black individuals. So again, it's really important that we, start looking within the Black community, trying to identify what are these profiles that we need to be focused on, since they are not appearing to be the same. Also, just a note, I did use dichotomous amyloid, and I used age, sex, demographic matched white cohort to Black, and our results were the same. So we really believe this. And I did look at global ancestry. I'm tending to go away from this and focus on local ancestry as I've learned more. Again, this is from a few years ago. But again, people who identify as Black in the United States have highly admixture ancestry between African ancestry, which is represented by the red stars, and European ancestry, which is represented by the blue stars. So each person has a measure of each of those 5 main global populations. So for example, just looking at the first column, you can see that first individual have African ancestry around 28%, but their European ancestry is about like 92%. And so you can kind of follow that through, and you can see they're completely inverse of each other. So I wanted to look at, rather than just race, how does amyloid measure to African ancestry? So again, amyloid SUVR is on the y-axis, and increasing African ancestry is on the x. And remember, higher amyloid is worse. So you can see, though, as African ancestry increases, amyloid decreases. So again, showing that this measure may not be exactly the same for these for Black individuals. And of note, this effect of ancestry is actually independent of APOE. And then just for time's sake, So selection bias, I think, is a major thing that I think a lot of us overlook. I did up until recently. Again, all of these tools have been validated and created in white individuals, applied to Black individuals. We know there's a lot of cultural bias that exists. So especially when you're coming into a clinical study like A4, you use a lot of neuropsychological assessments. And so I wanted to look at selection bias in the A4 study. And so we do see that if you were Black, you were more likely to screen out of the A4 study relative to white individuals at almost twice the rate. Why? Because they had too low of cognitive test scores. Now, the A4 study actually did have a norming procedure that they did for the neuropsychological assessment, but we can see that what is happening, that there's still Black individuals that are still screen failing, even with this norming procedure. While white individuals were more likely to screen fail because they had too high of test scores. So one thing that I would really like to see in the future, I really appreciate that the A4 data set, when they did publish the data, that they included all the people that, whether they were included in the study or not, so that we can look at this. And I would encourage other people with research cohorts to do this so we can start better evaluating this as well. I'm going to bypass this for time, but Black individuals more likely to have earlier brain aging relative to other groups. And then just to follow Dr. Kind, again, focusing on this health disparities research framework, because we are here for the NIA, from Dr. Hill, focusing on I'm now interested in really focusing on, social factors and how that impacts cognitive decline and dementia risk in Black individuals. And I really like this study from Dr. Glover, because this, again, Dr. Kind showed a really great way of how to incorporate the framework, but I just wanted to show another way in which you can focus on using the disparities research framework in relation to, for example, perceived stress. And something that I really want to mention is I'm really interested now in structural racism and discrimination. That's the current project that my lab is actually working on. And so I'm trying to focus on this and then related, also look at biological factors. And, experiencing racism over the lifetime, as you can imagine, can cause stress. Stress can cause changes in the body, which can actually cause these biological changes or affect biological processes. And so an example is a very recent study, I think just from January, from Dr. Zahadni, and she showed how discrimination is correlated with MRI measures of brain aging. So for example, at the top, we have hippocampal volume. And experiencing more lifetime discrimination was associated with having a lower hippocampal volume. And on the bottom, white matter hyperintensity volume, having more experiences of everyday racial discrimination, also associated with a higher white matter hyperintensity volume. And lastly, I definitely want to talk about research participation and health equity frameworks. Again, focusing on the topic of research right now is just really focused on European and white groups. We really need to do a better job at recruiting Black individuals and other groups, other racialized groups. I've often heard that it's too hard. And what we do is hard. Going through med school, going through a PhD, it's hard. But there's, we have to put effort into, I used to say an historically excluded group, but it's actually not historically excluded. It's still currently being excluded because the same efforts that we put into the other group is not being put into other racialized groups. And so it's really important, there are a lot of groups, again, such as Dr. Gilmore, Kopaski, sometimes I can say something, again, sorry, but have been putting out publications on suggestions on how to do it. Now, this is going to vary based on where you are, right? Black individuals from Los Angeles are not going to be the same as Black individuals from here, right? The type of research efforts. However, there are groups that are showing different frameworks in their research about how we can attempt to better do this. Don't really have a lot of acknowledgements for UCLA because I just got there, so it's really my lab is growing. I do have some other people, but again, I mainly showed work while I was at Stanford University with Dr. Beth Mormino, and of course acknowledging my key collaborators. Dr. Lisa Barnes has been a mentor for me since I was at Stanford and still is, and including this K1, which should be coming. And I just put older funding because exactly this K1 folks don't have grants yet. And then, of course, being a woman of color in science is really hard. And I am so thankful that I've actually been able to build a network of women of color in science. And this slide started with like 5 individuals and has grown to date. And I'm so thankful for them. I really honestly would not be here if it wasn't for the support and the mentorship, including not only research, but just mental health along the way. And so that's it.